Welcome you all, my dear students. So, we are watching a series of video lecture because in this pandemic COVID-19 situation, we are locked at home, but we have no other option. So, in this course of plant physiology and biotechnology, we have already discussed about several issues. I have uploaded several notes and lectures, and STGs are also uploaded. So I already covered one thing is recombinant DNA technology. Here we have discussed what is the recombinant DNA technology, how gene is transferred using vector. So now the vector, DNA vector and its types, several its features we'll talk about in this lecture tutorial. So first, what is a vector actually? Vector is a small DNA molecule capable of self-replication. So that means vector is nothing but a carrier. Carrier. What it carries a gene. Okay? Because your gene is not like the vegetables you are bringing from the market using a carry bag. When you are working with genes, you need to have vectors, the suitable vectors, so that you can carry the gene. So vector is a small DNA molecule capable of self-replication ability. That means it can replicate itself, cloning vehicle or cloning DNA. It can clone carrier of DNA fragment, carrier, it carries. Here are some examples. We'll talk about detailed examples and each of its characteristics later. Okay. So characteristics of common vector, DNA vector, self-replication of multiple copies, replication origin site. Origin site means where they starts replicating. Cloning site means you can easily insert your gene of interest. Selectable marker gene means suppose it's like a pie. Okay, in the vector you have the selectable marker which can express after cloning also. Okay, so small in size so that you can do the transformation experiment very easily. If it is bigger, then loss of nucleotides chances will be there. Low molecular weight. Once it is small is in size, that means it should be low in molecular weight. Easily isolated and purified. That should be the option. Easily isolated into host cell. Okay, so this is also a very important issue. And control elements should have promoter, operator, ribosome binding site. These are the new things I think to you. Promoter, operator, these are the some controlling DNA sequences which controls all the regulation of genes activity. So now we are talking about the types. What are the types of vector? Don't confuse with the example. We are talking about types. One first is the cloning vector and second is the expression vector. That's all. Is the two type vector, cloning vector and expression vector. So cloning vector means propagation or cloning of DNA inside a suitable host cell. Example plasmid, phage or virus. Obtaining millions of copies. Okay, and uses is genomic library preparation, preparation of probes, genetic engineering experiment. These are the uses. So what is a cloning vector? Cloning vector means such a vector where you can clone and make multiple copies of specific genes. Okay. So selection of cloning vector depends on several factors. How you will select, because there are lots of cloning vector based on your gene. How you will select the cloning vector? Your objective of cloning experiment should be on criteria. Then age of working, then knowledge existing about the vector, then suitability, reliability. So these are the things keeping in mind you can select the vector. So now the second type is the expression vector. One is the cloning vector, means you're inserting some gene to make it multiple copies. That is nothing but the clone. And second is expression vector, means you have inserted gene and you want to check the expression of the gene. Okay, whether this gene is properly functioning or not, properly working or not, that expression you need to check in expression vector. So what are the criteria for that? Express the DNA insert producing specific protein. They have prokaryotic promoter. This is very important. So in the expression vector should have prokaryotic promoter region. And ribosome binding site, it's an operating and controlling DNA sequence. Origin of replication is called ORI site, means where from the replication starts. Antibiotic resistant gene like NP2 marker gene, antibiotic resistant gene, so that you can check whether the other genes are working or not. Expression vectors with strong promoters. Strong promoter means who can regulate the activity of the gene. Inducible expression vector, eukaryotic expression vector, these are the some examples. So now it's all about vector and the target cell. Both the things are related to recombinant DNA technology. Don't forget, okay? So vectors are normally like the plasmid vector. I have told in the class for several times earlier. Bacteriophage, cosmid vector, yeast cloning vector, TIRI plasmid, okay? Tumor inducing, okay? So the vector and target cell means the vector should be inserted into the target cell. Target cell is normally E. coli is the bacteria, unicellular organism, or streptomyces bacteria you can use yeast, Saccharomyces cerevisiae, okay, transformation of the clone gene in higher plants also. But this is a very specific host cell, but normally we use bacteria and yeast. So now agent used as vector is the plasmid, bacteriophage, cosmid, artificial chromosome vector. In 1973, Cohen described first successful construction of a recombinant vector called plasmid PSC101 in E. coli is the host cell. So it is very important for MCQ. Note it down. So now, what is a plasmid first? Plasmid is the extra chromosomal DNA molecule. It has the ability of self-replicating, origin of replicating site, double-stranded DNA molecule, short sequence of the DNA, circular DNA molecule found in prokaryotes, means in bacteria. Okay, plasmid. And characters is the minimum amount of DNA, two suitable markers for identification. I will tell it later. Single restriction site, more restriction enzyme, size range 1, it should be kb to 200 kb, relaxed replication control restriction. So these are the characteristics. So you need to remember these things for the plasmid vector. Okay. So the plasmid vector and their types are: fertility plasmid can perform conjugation in the conjugation process in the bacterial gene transfer, and resistant plasmid contain genes that build a resistance against antibiotic or poisons. Okay. Cold plasmid contain genes that code for protein that kill harmful bacteria. So this picture you can see is like the recombinant DNA technology is the bacterial genome. Here is the bacterial plasmid. Here is the self-replicating cloned plasmid. So some examples of plasmid vectors are PBR322 is the well-known and very useful, commonly popular plasmid vector, and there are PBR327, PBR328, to 5, and PUC8 is also very important. Okay, so these are the examples. So now a little bit about the PBR322, as it is a very important plasmid vector. Okay, so cloning vector, it is a cloning vector and constructed plasmid already I have told, because it is made actually derived from E. coli plasmid, and its length is 4362 base pair BP. Okay, P denotes plasmid for B. Scientific Bolivar R is the Rodriguez is the scientist name, and 322 number is given to distinguish, because there are several plasmid, a series of plasmid, but 322 is the serial number. This plasmid shown the good activity, that's why they have selected. An ampicillin resistant gene derived from RSF, this is a one bacterial strain, and tetracycline resistant gene, origin of replication, two selectable marker. This is important. TTR and AMPR. TTR, AMPR, these two selectable marker. This is useful because once the cloning experiment is over, you need to check whether your gene is properly functioning or not, along with these two selectable marker, these two selectable gene, because it's already there in the plasmid. So if you check, if you can observe these two markers are functioning, so your gene, inserted gene should be functional. So this is a typical picture of a UC3T 322 PBR322. Okay, so PBR322 is the total size, is the amplification, TET is the gene. Okay, and here is the ori site, means from here origin of replication should start, and these are the restriction sites. Restriction site means here, suppose in 3 or EcoR1 is the restriction enzyme, you can cut it here and you can insert your gene here. Or if you have BAM H1 restriction site, so you can cut it here, insert gene here. Okay, then transform into the bacterial cell. So is the all about plasmid DNA of PBR322. This is the another PBR327 is derived from PBR322, and is the 30 to 40 copies can be derived at a single time. Expression, plasmid, amplification resistant genes is the common features. Okay, so it's PUC8 is also important. PUC8 is the total length is 2700 base pair, and LAGZ gene is derived from E. coli LAGZ. In the recombinant DNA technology, you can see there are three genes, lag XYZ. Okay, so today is for this one is enough, I think. So we can continue in the part 2. Thank you for watching.